Hello everyone, I'm Chino Bervias Zapanta, and for today's video, I'm going to continue discussing the five senses. There are only two remaining senses that I'm going to discuss because the three senses was already discussed by Mr. Bert Noel, and those two senses that I'm going to discuss are the sense of taste and sense of smell. So without further ado, let's start. So sense of taste, or what we call as gustation, is a sense that develops through the interaction of dissolved molecules with a taste buds. Currently, there are five modalities or submodalities taste that being recognized, including sweet, salty, bitter, sour, and umami, which is the savory taste or the taste of protein. Taste is associated mainly with the tongue, although there are taste or gustatory receptors on the palate and epiglottis as well. The surface of the tongue, along with the rest of the oral cavity, is lined by a stratified squamous epithelium. In the surface of the tongue are raised bumps called papilla that contain the taste buds. So, what are those small bumps on the top of our tongue? They are actually called papillae. Tongue is covered with 1,000 of small bumps called papillae, which are very visible in our naked eye. Within each papilla are hundreds of taste buds. You know what? There are three types of papillae. First, we have the valate papillae, or what we call as the circumvallate papillae. The function of this is to detect better taste. And also we have the foliate papillae. So the function of this one is to perceive taste. And also we have the last one is fungiform papillae, which is the function of this is to detect the flavors as well as the temperature and the touch of the particles that make up the food. So when we eat, those chemicals that coming from different food, it enters to our papillae and reach the taste buds. So these chemicals or tostans stimulate specialized gustatory cells inside the taste buds. So it activates the nervous receptors. So these receptors send signals to fibers of the facial. So glossopharyngeal and vagus nerves, those nerves carry the signals to the medulla oblongata, which relays them to the thalamus and cerebral cortex of our brain. How do we perceive this taste as flavors? Food contains chemical substances which are released in the mouth and detected by sensory cells. Sensory cells are located on taste buds or papillae, which are found scattered all over the tongue. In particular, the fungiform, valate, and foliate are the papillae responsible for detection or different tastes, just like what I've said for those three types of papillae. Once it activated, the sensory cells transmitted messenger substances, which is to activate further nerve cells. These nerve cells then pass information for a particular perception of flavor on our brain. Through this process, different flavors can deeply influence our mood, emotions, and memories. The taste of like ice cream, chocolate, can trigger the happy emotional regions of your brain, our brain, bringing a new meaning to the term comfort food. So now let's have taste sensation. So we have five submodalities as I previously mentioned earlier. Is that salty allows modulating diet for electrolyte balance, and also sour, which typically the taste of acids, and also we have the sweet, which usually indicates energy-rich nutrients, and also bitter, which allows sensing of diverse natural toxins, and lastly is umami, which is the savory taste, or what I said earlier, that the taste of amino acids. So example of this is kanang mga mint bread or mga aged cheese. So these different taste of sensation are currently recognized is that salty is simply the sense of Na plus concentration in the saliva. So as the Na plus concentration becomes high outside the taste cells, A strong concentration gradient drives their diffusion into the cells. This depolarizes the cells, leading them to release neurotransmitter. And also, the sour taste is transduced similar to that salty one, because except that it is a response to the H plus concentration released from acidic substances, those with a low pH. Instead of response to an Na plus, like for example, orange juice, which contains of citric acid, will taste sour, diba? As lumikit siya, because it has a pH value of about 3 plus. Of course, it is often sweetened so that the sour taste is masked. The concentration of this is that the hydrogen ion increases because of ingesting acidic compounds, which the depolarization of specific taste cells increases. And also the other three tastes, which are sweet, bitter, and umami, are transduced through G protein coupled cell surface receptors instead of the direct diffusion of ions, like we discussed with salty and sour. The sweet taste is the sensitivity of taste cells to the presence of glucose dissolved in our saliva. Molecules that are similar in structure to glucose will have a similar effect on sensation of sweetness. Other monosaccharides such as fructose or artificial sweeteners like mga condensed or NutraSweets or whatsoever, that will activate the sweet receptors as well. So the affinity for each of these molecules varies, and some will taste sweeter. It's tastes sweeter than glucose because they bind to the G protein coupled receptor differently. And also the bitter taste. The bitter taste can be stimulated by a large number of molecules collectively known as alkaloids. Alkaloids are essentially the opposite of acids. They contain basic in the sense of pH, nitrogen's atom within their structure. Most alkaloids originate from plant source, with common example being hops in beer, tannins or in wine, tea, aspirin, and similar molecules. Coffee contains alkaloid also, and it is slightly acidic, with the alkaloids contributing the bitter taste of the coffee. The taste known as umami is often referred as the savory taste. The name was actually created by the Japanese researcher who originally described it like sweet and bitter. So it is based on the activation of G protein coupled receptors, in this case by amino acid, especially glutamine. Thus, umami might be the considered taste of protein, and it is most associated with meat that containing dishes. Taste buds and taste receptor cells. The sense of taste is mediated receptor cells, which are bundled in cluster called taste buds. Taste receptor cells sample oral concentration of a large number of small molecules and report a sensation taste of centers in the brain stem. In most animals, including humans, taste buds are most prevalent on small pegs of epithelium on the tongue called papillae. Taste buds are small organ located primarily on the tongue. It allows you to experience tastes that are sweety, salt, sour, and bitter. You know what? The adult human tongue contains between 2,000 and 8,000 taste buds, each of which are made up of 50 to 150 taste receptor cells. Taste receptor cells are responsible for reporting the sense of taste to the brain. Taste receptors or taste buds contain the taste receptors. The taste receptors are located around the small structure known as papillae, found on the upper surface of the tongue. The taste receptor cells send information to detect by cluster of and various receptors and ion channels to the gustatory areas of the brain. In detecting the five elements of perception, which is the saltness, sourness, bitterness, sweetness, and umami. I'm going to show you now a short video with regards to sense of taste. So, in order for you guys to know more about sense of taste, so watch it.
Taste, or gustation, is a special sense that helps us evaluate what we eat or drink. Basically, certain food molecules dissolved in the saliva activate taste receptor cells in the mouth. These chemical stimuli are then converted into electrical signals and sent via several nerves to the brain, where they are interpreted as tastes. Taste receptor cells are organized in groups called taste buds, which are present most abundantly on the tongue but also on other parts of the mouth. On the tongue, taste buds are located on small visible bumps called papillae. There are different types of papillae on different parts of the tongue, but the taste buds that they contain are all similar in structure. Each taste bud is composed of about 50 taste cells with microvilli at the top projecting into a pit called the taste pore. This is where taste molecules bind to their receptors. Taste cells synapse with sensory nerve fibers at the base of taste buds. There are 5 main types of taste receptors corresponding to 5 major taste sensations in humans. Salty receptors detect sodium and salt. Sweet receptors bind to a number of sugars and sugar substitutes. Sour receptors activated by acids. Umami, or meaty taste, elicited by amino acids, in particular glutamates. And bitter taste associated with spoiled foods, natural toxins, and substances such as quinine, caffeine. Each taste cell has receptors for only one type of taste, but a taste bud is typically composed of a variety of cells detecting different tastes. All primary tastes can be perceived throughout the tongue, but some regions are more sensitive to a certain taste than others. Each of the primary tastes can be elicited by not one, but a number of chemicals. In addition, the thresholds for detection differ among chemicals that taste the same. Sweet and salty substances generally have high thresholds. They must be present in large amounts to be detected. Bitter compounds typically have very low thresholds. We can taste them at very low concentrations. Taste buds at the back of the tongue are especially sensitive to bitter substances, which often trigger rejection responses such as gagging and vomiting, an attempt to avoid ingestion of toxins. Some people are super tasters. They have more taste buds on their tongue and are therefore able to detect certain subtle tastes at very low concentrations. Depending on the type of taste sensation, binding of taste molecules to their specific receptors results in depolarization, or activation of G protein and second messenger signaling in taste cells. In either case, the activated taste cells release neurotransmitters which generate actions potentials in sensory nerve fibers. Depending on the location of the taste buds, nerve fibers carrying taste signals move along cranial nerves 7, 9, or 10 to the solitary nucleus of the brainstem. From there, second-order neurons project to 2 destinations: the thalamus, where they synapse with third-order neurons which continue to higher cortical taste centers, and the hypothalamus and amygdala, where they trigger autonomic reflexes such as salivation, gagging, vomiting, and provide input for regulation of eating behaviors.
So that's all for the sense of taste. So let's now proceed to the last sense, which is the sense of smell, or what we call as the olfaction. So the sense of smell or olfaction is the special sense through which smells or odors are perceived. The sense of smell has many functions, including detecting the desirable foods, hazards, and pheromones, and play a role in taste. In human, it occurs when odor binds to a receptor within the nasal cavity, and it's transmitting a signal to the olfactory system. Sense of smell or olfaction start with specialized nerve receptors located on cecilia in the epithelium at the top of the nasal cavity. When we sniff or inhale through the nose, some chemicals in the air bind to these receptors. It triggers a signal that travels up a nerve fiber through the epithelium and the skull bone above to the olfactory bulbs. Olfactory bulbs contain urine cells. It contains urine cells bodies that transmit information along the cranial nerves, which are extension of the olfactory bulbs. They send the signal down the olfactory nerves toward the olfactory of the area of cerebral cortex. The molecules that activate the sense of smell, the technically name is olfaction, are airborne. They enter the body via the nose and mouth and attach to receptor cells that line the mucous membranes far back in the nose. In humans, there are millions of such cells altogether, but only several hundred, like 400 is good estimate, different types of olfactory receptors. One thing that makes the olfaction unique among the senses is that its receptor cells are themselves neurons. Each olfactory receptor cell has filaments that what we call as cilia, or cilia, not Cecilia di ay, with receptor designed to bind to specific molecules. Like all neurons, the cell also projects a thicker fiber which called the axon. The axons come together in olfactory nerve and go directly to the brain. I will show you a short video of a sense of smell so that you guys
The olfactory
can understand
system
more
is
about,
responsible for the sense of smell or olfaction.
So, watch it.
Basically, airborne molecules emitted by an odorant source are detected by olfactory sensory neurons located at the roof of the nasal cavity. These neurons convert chemical stimuli into electrical signals and send them via the olfactory nerve to the olfactory bulb, then to the brain, where they are interpreted as odors. Odorant molecules are first dissolved in the mucus secreted by the olfactory epithelium, which guides them to the cilia of olfactory neurons. This is where odorant molecules bind to their receptors. Each neuron expresses a single type of protein receptor. There are only about 400 different receptors in humans, but they are used in a combinatorial way such that one odorant can bind several receptors, and one receptor can bind several odorants. This enables the olfactory system to recognize an enormous number of odorants. Odorant receptors are G-protein coupled. Upon binding to the odorant, a signaling cascade is activated, leading to membrane depolarization. When the olfactory stimulus is strong enough, action potentials are generated and conducted along the axon to the olfactory bulb. The axons of all olfactory sensory neurons form the olfactory nerve, also known as cranial nerve 1. In the olfactory bulb, these axons synapse with second-order neurons, the mitral and tufted cells, within structures called glomeruli. Each glomerulus receives axons from sensory neurons that express the same protein receptor. The second-order neurons are stimulated by sensory neurons, but they also receive inhibitory feedback from the cerebral cortex. This means an odor can be perceived differently under different circumstances. For example, the smell of food is more appealing when one is hungry and is less so when one is full. The axons of mitral and tufted cells form the olfactory tracts, which project directly to the primary olfactory cortex. The primary olfactory cortex is not one, but several cortical areas located on the base of the frontal lobe and inferior surface of the temporal lobe. These primary regions then project further to some other areas of the brain, mediating different aspects of odor recognition and response. Because olfactory neurons are exposed directly to the noxious external environment, they are replaced more often than other neurons. Stem cells in the epithelium differentiate into new olfactory neurons, whose axons grow along the existing axons to the olfactory bulb. Any factors that destroy all olfactory neurons at once would result in a permanent loss of sense of smell, a condition known as anosmia. Illnesses that cause inflammation of the nasal mucosa may lead to transient anosmia. Loss of smell also affects the taste experience, as taste and smell are the two aspects of flavor. The ability to smell decreases with normal aging, but anosmia is also an early sign of several neurodegenerative disorders. Because epileptic seizures often originate from the brain area associated with the olfactory cortex, seizures are often preceded by hallucinations of disagreeable odors.
So that's all, guys. Thank you for listening, and God bless you.